All right. All right, we seem to be on here. Let me...
Oh, by the way, my mic is hot, right?
It is.
Okay, so then I have to shut up.
Okay. While people are entering, give them another minute. I'm seeing participants trickling in right now. We are here in San Francisco at a beautiful podcast room that was specifically equipped to allow for these beautiful webinars to take place in person. And now for the first time, last two webinars were over the air, over the internet, but this time we're sitting right across each other. Now, let me get started here. So we have everybody, or some of you, joining for our new version of the webinar that is hosted by Modality. Today we will talk about audiovisual biomarkers in CNS trials, trials of the central nervous system. And our guest speaker is Vikram Ramanarayanana, Dr. Vikram Ramanarayanana, the Chief Science Officer of Modality. And he is not only Chief Science Officer of Modality, but he's also an adjunct professor, associate adjunct professor at the University of California in San Francisco, UCSF, also right here in the city of SF. You know me, I'm moderating as usual. I'm DSO, CEO and founder of Modality. So today we have two modaliteers in the room, while before, of course, we had external speakers as well. That's going to be extra fun today as we dive deeper into what audiovisual biomarkers mean. This is what we want to do today. Quick introduction of Vikram, but also of the company, just a recursor. We will have him actually present a little bit in more detail a recent paper that was published in the Journal of Speech, Language, and Hearing Research called Multimodal Technologies for Remote Assessment of Neurological and Mental Health. This will give us some background information about the conversation which we have afterwards, diving a little bit deeper, double-clicking on some of the topics that will be covered there before we close in 45 minutes. Again, as usual, we appreciate audience interactions, and any questions you may have, please put them into the chat as well. We had really many of such last time, and so we would love to continue this flow, and Vikram is really happy to answer those. So without further ado, let me just go through a quick round of really high-level introduction of what we talk about here. We talk about clinical trials challenges, specifically to answer the question whether drugs are working. Very often this question cannot be answered, which is one of the reasons why clinical trials fail. In CNS conditions like Alzheimer's, for example, more than 90%, more than 95% of trials actually fail. So that's something we want to address here. Inadequate assessments is one of the major causes for this, and that's really because today, as you know, most of the assessments happen in the clinic by having interviews between neurologists and patients who fill out these surveys, such as the UPDRS, MDS UPDRS, or ALS FRSR scales that are typically used. We talk about multimodality today, audiovisual biomarkers today. This is an example here. Modality uses TINA, a virtual guide, that interviews patients, and we can tell from what patients say and do in response to these interview questions by analyzing people's facial expressions, body movements, the things they say, for example, speech and language, but also cognitive aspects, even the specific symptoms they're mentioning in response to questions that TINA has. And this is something that is being provided on people's own devices, usually at their home. You see here, laptops can be used, smartphones, iPads, anything that's web connected for that matter. And again, Vikram is going to talk a little bit more about TINA specifically, but also more generally about the entire space of multimodal assessment technologies. We are being used usually by biopharma sponsors, biotech companies, big pharma companies, but also by the federal government, who sponsors a lot of studies as well. You see here some examples, the Veterans Administration, NIH, but then also J&J, some big pharma companies and others. Just briefly, Modality very scientific entity. We have over 105 publications. Actually, Vikram is the lead of the research team at Modality. We have something like 7 PhDs that are working with Vikram on an ongoing basis to produce these publications. 38 studies ongoing. We are almost at 150 clinical sites today. So these are mostly for interventional trials in 18 countries and have touched the lives of 80,000 participants so far. You see here a little overview of the individual studies that we've been engaged with. And now, without further ado, let me introduce Vikram, our Chief Science Officer. He, as I mentioned, also adjunct professor at University of California in San Francisco, specifically working on speech science over there. And we met something like 10 years ago at ETS. Vikram was senior research scientist there and manager of the San Francisco office at that time. And so before that, he got his PhD at the Reproducible School at the University of Southern California, USC in Los Angeles, in electrical engineering. That's also something that we share as our background. He has over 170 scientific publications, many of them together and many of them at Modality, actually. And so now I want to hand over to Vikram because he's going to introduce you to some of the essence of his scientific paper, which will form the basis of our conversation today. Thank you, Vikram. We'll stop sharing because Vikram has a few slides prepared for you.
Wonderful. Thank you, David. That was a wonderful introduction. Thank you so much for that. Let me quickly share my screen so that we can get started. All right. So what I'd like to talk to you a bit about today is this notion of audiovisual biomarkers in CNS trials. So the central question that I'm going to be asking is, why capture digital biomarkers from multiple modalities, right? Audiovisual, why multiple modalities? In other words, why multimodal, right? And in order to answer this question, I want to make a distinction between two uses of the word multimodal, or two uses of the word modality. So we've heard of modalities in terms of modalities of signal recordings or signal sources, and we've heard of modalities of health information. And I want to make this distinction because this is going to make a point of why multimodal. So for example, I can take a piece of text, like we have chats from participants. This can tell us two different kinds of health information modalities, which are embedded within. One is just the natural language, the pragmatics, the linguistics, the syntax, the morphology of their language. And the other is how they're interacting with people, the dialogue. We can extend this. So if you record audio from people, you can record audio of their speech. It could be their respiration. It could be coughing, other non-speech sounds. You also capture natural language inherently because of speech. You're able to capture interactions through speaking, through dialogue as well. And then you extend that to video. You can extract a lot of health information from one video signal. For example, from the same video that you see of me, you can record my orofacial movement. You can record my limb movement, like if I'm tapping my fingers. You can record my eye gaze. And you can even record cardiopulmonary information regarding blood flow, heart rate, things like that. All of this from that one signal modality video. Not just that, all of these modalities tell you something about cognition. How I'm thinking, what's my reaction time to doing certain events, how quickly is my recall, and so on. Each of these health modalities tells me something complementary to the health state of a person. And then when we take this a little bit further, we also have sensors and peripheral devices like tablets, computers, for example, that allow us to capture a range of these modalities as well as a couple of others. An example is portable EEG, for example. So the idea that I'm going to propose is that you have 4 improvements at least by combining these various modalities, including improved clinical interpretability, improved assessment performance, improved user experience, and improved technology robustness. And I'll go into specific examples of what I mean by all of this. But before I do that, I wanted to mention that the modality assessment inherently embodies this philosophy. And that's, by the way, why we're called modality, if it isn't obvious already. So we record text, audio, and video, signal sources, but we extract a plethora of information modalities, health information modalities from that. So let's go into the improvements. Let's talk about interpretability. The idea of speech being a biomarker has been explored in and proven in multiple studies in the literature. And this is just one example that comes from a paper that I co-wrote with authors from MIT and MGH. And this actually synthesizes decades of research that demonstrates that in any neurological or physiological condition, speech impacts multiple domains, including cognitive, linguistic, affective domains, motoric, and anatomical. And we can extract biomarkers from speech that are representative of each of these domains of health. So examples include pauses or articulation rate. We can also, for example, extract movements of the lip and jaw that capture, for instance, motoric movement. And so this multifaceted nature of speech allows us to get a lot of interpretability into different domains. That's exactly the case in terms of if you look at different diseases. So for example, if you look at the motoric domain, you see Parkinson's disease, ALS, Huntington's disease. Each of these, they're biomarkers that are speech biomarkers that capture specific aspects of them. Progression, early diagnosis, and so on. The same goes for cognitive diseases like Alzheimer's disease or linguistic conditions like PPA, primary progressive aphasia. We have neurodevelopmental disorders like autism, affective disorders like depression, schizophrenia, and so on. We can extend this to text or natural language, because natural language, as we all know, is not just what you say. It's about the syntax, the semantics, the phonetics, the phonology, morphology, discourse. There are different levels of these that are impacted in different diseases. Video adds an extra dimensional of versatility, because now you can capture, as I mentioned, motoric aspects. You can capture eye gaze, which is useful in diseases like neurocognition. The motor function is extremely important, say something like Parkinson's disease, where the finger tapping test is actually a part of the UPDRS. And you can also capture emotion through facial expression. So all this is great. We've seen how each of these individual modalities are useful for clinical interpretability, but let's put them all together and see what happens. In a recent paper, we showed that if you look at capturing a holistic picture of mental state of a person, and this was published in Frontiers in Psychology, we see that speech features actually perform the best for depression. Facial features perform the best for anxiety detection. And text or natural language-based features perform the best for suicide risk. So in other words, the content of a person's speech, what they're saying, is really most important when you're talking about suicide. But when you're looking at anxiety, it's something more inherent. It's more about how facial expressions or reactions to people's utterances or conversations are observed. And depression is something that you see more in speech. It's not that you don't see it in the other modalities, though, because what we see is, and this is the second advantage, that you combine all these modalities and you actually get the best performance for assessment. And this is actually one of the key reasons why we say, combining modalities is really, the most tangible thing to see in terms of performance. So not only do you get the best performance in terms of depression, you see this across the board in multiple conditions. So here's an example from schizophrenia, where if we are looking at the performance accuracy, the diagnostic accuracy of a test in detecting schizophrenia versus healthy controls, you overwhelmingly see a 90% area under the curve accuracy, which is a way of determining diagnostic accuracy, when you're combining modalities, in this case, speech and facial for this example, versus the individual modalities alone. We can take this a step further. And by the way, this is an example from ALS. you can actually combine these modalities, each of these different features, into a single index that not only improves statistical power and accuracy of your test, it improves statistical robustness, because it turns out that these modalities some of these modalities are complementary in that they provide different information to boost the accuracy. Some of these are redundant in that they provide the same information. So for example, if one of my biomarkers is not performing well or is noisy, it's made up for it by information from the other biomarker. And that's the intuition for why this is noise robust and has good properties. And so for many clinical trials where you might just want a single endpoint, this actually combines all of these into a single number. Let's move on to the third advantage, technology robustness. So we've talked about performance, we've talked about clinical interpretability, but it turns out that combining modalities actually makes the technology that you're using to deliver these assessments way more robust. A simple example comes from this idea of voice activity detection. So any technology which involves speech requires you to identify which portions are speech and which portions are non-speech, or silence or noise or whatever. This technology is called voice activity detection. Now, it stands perhaps as a simple matter of intuition that if I just use the speech to determine when the person is speaking versus when the person isn't, especially in the presence of noise, versus if I have a video of the person's lips moving up and down, I will get a better sense of when that person spoke or when did that person not. And this is the idea of technology robustness. So it's multifaceted. We can take this a step further. The idea of combining both what people said and how people said it through video, for example, can allow us to do better dialogue management. And this is actually work that David and I both did actually at ETS that demonstrates this very clearly, that people tend to speak more, they're more engaged with dialogue systems if you actually combine these complementary but doubly informative modalities. And last but definitely not the least, there's this idea of immersive user experience, or UX, and the fact that using all these technologies, especially when you combine them with the modality of dialogue in an interactive virtual agent, we also call this an embodied conversational agent in the computer science literature. It improves retention and user experience. We've looked at it across the board. This has been shown in young populations, like with children with autism, elderly populations, people with dementia, that you get better data and more engagement from participants, and they're more motivated. We've also seen this with modality, where we found that participants felt more engaged with an animated teacher, where she blinks and smiles at you, and they found her more intelligible even than when this was not the case. And moreover, if you look at people who had mild to moderate depression versus, or mild to no depression, and I apologize, versus moderate to severe depression, we see that the user experience was equally good of this kind of system. So there's no differentiation between people who are experiencing a certain mental health condition, for example. In fact, what was funny was that people with moderate to severe depression actually preferred non-human voices or synthesized voices, which tells us that TINA and technologies like embodied conversational agents allow you to engage in interaction with participants without giving the feeling that they're being judged. So this is actually an advantage when it comes to combining all of these modalities together into one single solution. So this is my last slide, and that's what I want to leave you with, the central message of the paper, and I'd encourage you to read it more. There's a lot more detail about the various nuances, the references to papers that I'd encourage you to read. But the inherent take-home message is that by combining all these modalities into one single system, especially one that includes a dialogue agent, an embodied conversational agent, you will get better clinical interpretability, better performance, better technological robustness, and last but not least, better user experience. And with that, I will stop sharing, or I'll keep this slide up maybe, so that, because it's a good perhaps basis for the conversation between David and me.
Absolutely. And just to remind the audience, please feel free to ask your questions through the Q&A feature of Zoom here, the Zoom webinar. because we are getting already some questions coming in right now. But before we go into those, I want to ask a very high level question, and something that we are hearing really frequently from our customers. And is that technology going to help them answer that very simple question: is my drug working? So what would you think?
So the answer to is my drug working is a difficult question to answer, but we can answer a question: how is my drug working? Because the is my drug working is a very complicated question that is based on a lot of factors, pharmacokinetics and subject specificity, recruiting. But what we can say is how is my drug working? Because we are able to measure multiple modalities of health information, each of which, like we showed the example of speech, where you're impacting domains of cognition, you're impacting domains of motor function, you're impacting domains of emotion or affect. So we can capture with specific biomarkers how your drug is impacting each and every one of these single modalities. And so we can tailor biomarkers for specific mechanisms of action of your drug. So that way, take the example of something like a schizophrenia drug, right? So we have both primary negative symptoms as well as secondary negative symptoms. We can break these down. We can kind of see, okay, what are the motor effects of your drug unwanted, for instance, tardive dyskinesia. We can look at, how it affects things like alogia, which is the inability to produce large amounts of speech. So does it actually improve how well you speak? We can test these things with specific biomarkers in a quantitative manner. So the nice thing about this is it's also not qualitative. It's not subjective. It's not something where it requires 2 different or 3 different neuropsychologists to come into the room and say, okay, I think that I would rate this person's expressivity on a 3 out of 4. And another person comes and says, 2 out of 4. How do you know that? Because these are discrete categories. Instead, we have a continua of biomarkers, which can tell you how things vary continuously in time.
Yeah, it's an interesting example that you're giving with schizophrenia. Just a couple of days ago, or yesterday actually, the announcement that KarXT, the BMS drug, is now approved by the FDA as one of the drugs in schizophrenia. I think it's been a couple of decades since a new drug hit the market, and the advantage of that one, of KarXT, is it provides similar efficacy in terms of the specific schizophrenia symptoms, but it provides also a better way or less strong side effects that are being reported. And now the question is really, as you pointed out, is my drug working? It's probably not it's
a tricky question, right? So instead, what we can do is actually stratify. We can kind of say, okay, how is my drug working in terms of capturing anhedonia? How is it working in terms of capturing alogia? How is it capturing in terms of other kinds of motor effects, tardive dyskinesia? Those are the kinds of things that we can pinpoint. Like, for example, we can look at facial movements. We can look at specific aspects of cognition that you get through text features. You can look at the content of the speech. So all of these are specific targeted biomarkers that allow you to capture different aspects of how your drug is working. So I think that is the promise of getting something which is truly multimodal, as opposed to something which is just movement or just speech. All of these packaged into one and helping us understand what are the relative degrees to which it affects each of these categories.
That makes sense. And you mentioned quite a number of different ways how these multimodal technologies can extract things with, for example, language sort of endpoints. You mentioned facial movements. You mentioned, for example, in the depression and anxiety and suicidality study that many different aspects play different roles in these conditions. Now, how do you go about validating these? And is that very difficult because there's so many things you need to look at?
It's not simple, but there is a nice principled framework, and there's a set of principled frameworks for this that are well adopted today. which is also something which is inherently true in all of the papers and all of the studies that Modality does. It's called the V3 plus pipeline that is actually proposed by the DIME, the Digital Medicine Society, among other people. So it's a consortium of group of people. Standard in the field. V3 plus stands for verification, analytical validation, clinical validation. And those are the three primary components of it. Verification is basically talking about, are my signal sources, is my signal being recorded with high fidelity? Analytical validation is saying, whatever my automated algorithm provides, is it as accurate as what a human would provide if that human were to do the same task? So for example, if I were computing speaking rate from speech, if I had an automated algorithm do this, how accurately is it doing it relative to how a human would count the number of words and divide by the number of seconds or divide by the number of minutes, right? That's analytical validation. And then you have clinical validation, which is essentially saying, what is the clinical utility of this biomarker? How clinically useful is it? And this can be measured in a number of ways. You can look at, for example, how correlated my biomarker is or my endpoint is with different clinical gold standards of interest. For ALS, it might be the ALSFRS-R. For Parkinson's disease, it might be various aspects or subsections of the UPDRS. For schizophrenia, it might be the PANS or the BNSS. There are many scales depending on what you're specifically looking at. But the idea is getting at, can I show that my biomarkers are clinically meaningful? The idea of clinical meaningfulness can actually be taken a step further, because the idea is, of course, for clinical meaningfulness, you want to look at, how a patient feels, functions, and survives. For this, we also have an instrument called the PROP, which you mentioned right off the bat in the beginning, where we are explicitly asking patients in their own words what bothers them the most about their condition and how it bothers them or how it affects them by, affecting their daily functioning. And we explicitly capture this in addition to all our biomarkers.
It's really interesting you say that because clinical meaningfulness is something that's coming up in many of our conversations with customers and partners. And so I was wondering about the V3 pipeline that you just mentioned that goes about verification, analytical, and clinical validation, whether the clinical validation piece is actually the same as showing clinical meaningfulness, or whether there's still another step to be taken.
There is another step to be taken, right? So it's a composite. So it's clinical meaningfulness is looking at objective, for example, it perhaps encompasses all of these things. I think our approach to it is looking at a unified approach, just like with the way we're doing with multimodality, combining information from all of these sources. We want to demonstrate both clinical utility, meaning it is representative of the current clinical gold standard, sometimes even more sensitive than the current clinical gold standard. Like, for example, in the case of ALS, where we've shown that our biomarkers are actually more sensitive. They pick up change faster than your traditional ALSFRS-R. We want to show that, but we also want to show that it actually has or is capturing a notion of clinical meaningfulness in that it is capturing at least patients' function, how they feel, function, and survive. So these biomarkers definitely capture aspects of how they function, and the PROP allows us to capture how patients feel and survive. So putting all of this together allows a more holistic and a more complete description of essentially the answer to your first question, which is, how is my biomarker working?
Yeah. How is my drug working or affecting
how is my drug working?
The patient? And it's really interesting question, because also, as you go through these different stages of trials, like you have these phase 1b proof of concept studies where you really want to find out, usually, of course, whether the drug is safe. That's mostly what happens there, or what's the optimal dose, dosage for the drug to be administered. And then in phase 2 study, you really want to identify already your markers of efficacy. Right. And then, of course, at the end, for getting approval, you need usually phase 3 study that you run through. And the stakes are different, and the questions are somewhat different. Except for that, the general is my drug or how is my drug working is basically a common theme even throughout those. But of course, in the phase 3 study, clinical meaningfulness is really important, because that's what the FDA wants to see, or the other regulators want to see. Not only that there's statistical significance, but also clinical meaningfulness. While before, in these earlier stage studies, it is often or sometimes not really necessary yet.
That's right.
Because the cohort size is also small and so on. So there it is really powerful to have even changes that you mentioned. I think with ALS, for example, you can identify changes significantly earlier than the regular scales.
That's right.
And that's something that is meaningful already in earlier stages to actually demonstrate this, because then you get confidence in that the drug is making a difference.
Exactly right. Yeah.
I'll put it that way. That's really interesting. And I was wondering, as you're talking about these different types of technologies, whether it is language processing, for example, you mentioned the prop. People describe in their own words what their most bothersome problems is. And by the way, for the audience, if you want to learn a little bit more about this, we had a webinar last time with Andrew McGarry last month, and that was diving deeper into what the prop is and how clinical meaningfulness is exposed there. But in the essence is that Tina asks patients to describe in their own words the most bothersome problems they're experiencing with their disease, and so they then answer in the level of detail they feel comfortable about this question. And usually they answer in pretty much detail.
Extraordinary detail.
Yeah, in extraordinary detail. They speak for 3 minutes or so to this one question, and then we allow them to speak more, and they continue.
That's right.
That's usually the case. So our participants, they love to talk to Tina to provide details, and that's of course for us an opportunity and a challenge, both.
That's right.
Right? Why is it opportunity and why is it a challenge?
Well, lots of opportunities, right? Because we get to understand both, what bothers them, because it's directly clinically meaningful, right? But then we kind of look at a range of information from this, because again, we are recording their speech. It's not that they're writing down what bothers them the most. They're speaking. We're seeing their facial expressions. We are looking at their speech. We are combining all of this information together, right? So and of course, in order to do this, by the way, this may not be apparent to the audience, but we actually use the latest and greatest in AI. We use the power of large language models. We use, all of the technology, for example, that goes into ChatGPT, a lot of those kinds of technologies, similar technologies, in order to pull out clinically meaningful symptoms that would make sense to a doctor if they were annotating this. So this is one side of it, of course, but then the other side is, it's large amount of data. How do you actually parse through this amount of data? You have to, and again, so our answer to that, and then if you, some of it was covered in the last webinar with Andrew McGarry, but the idea is that we are, again, looking at something more holistic. We are using the latest and greatest in technologies, large language models, but we also are making sure that all of these are infused with clinical knowledge, with neuropsychologists, neuropsychiatrists who have curated the initial version, for example, of this prop for Parkinson's disease. So this is how we tackle this deluge of data that we get, which, by the way, is, even it's a large amount even for an active assessment, which is what we are, as opposed to other kinds of assessments which generate way more kinds of data. But yeah, so it's interesting, but I think we are at this right cusp. We are rightly positioned at Modality because we have the right expertise. We have interdisciplinary expertise in not only the AI machine learning space, but also the clinical neuroscience, neurology space, which allows us to kind of fuse these things together and tackle these challenges at scale.
And I definitely want to go deeper into this question because it's a really interesting one. But we have a number of questions from the audience, which I want to prioritize here. The first one is, how effective are these methods in early stage detection? And what strategies do you employ to obtain reliable ground truth data? That's a really interesting question. When focusing on early detection, this is crucial for ensuring the validity of the results, right?
Yes.
So what do you think?
Yeah, that's a great question, Sudarshan. Hi, nice to see you. How effective are these methods? They are effective to varying degrees. It really depends on the disease that you're talking about. So, for example, let's take something like limb onset ALS. So we've done some work in this. We've shown that we are able to do a good job with these kinds of multimodal features, especially with facial. You tend to find out a little bit more on facial features than in speech features with limb onset. But it's again comes with a caveat. The diagnostic accuracy is lower, and of course, it's a harder problem, right? How do you detect it early enough? We've also seen with certain speech biomarkers like canonical timing alignment, which captures the essential the mismatch in temporal alignment of your phonemes, or the duration of your phonemes as you kind of progress in the disease. We've seen that is sensitive to early diagnosis for ALS. But it's tricky, right? Because the second part of your question is, what's your ground truth? And the short answer is, typically it tends to be the current clinical gold standard, which happens to be the ALSFRSR. And the ALSFRSR, as we've all seen, has its own set of challenges because it is coarse, it is not as sensitive. So it's kind of a chicken and egg problem. So one thing we've actually done in the recent past, and it's not just us, even our collaborators at Everything ALS have actually looked into this idea of listening effort, where listening effort is something which is a little bit more clinically meaningful. It basically measures how much effort it takes to understand or listen, to someone who has ALS speech. And you just rate it on a scale from 0 to 100. And this is a more continuous valued measure. This is something that we've had a little bit more interesting results with. And it's perhaps a little bit more sensitive measure to ALS progression. And we've seen that this canonical timing alignment measure that we automatically extract actually captures that and correlates with that very well. So hopefully that answers that question.
Yeah, I want to add to this very briefly, because it's not only about ALS, but there's other diseases.
There's several other
like,
conditions.
for example, with mild cognitive impairment, which is an early
That's right.
stage, potential early stage for dementia, but it is actually not dementia because it's not yet diagnosed. So
That's right.
you are diagnosed with MCI, which is really difficult actually to do to begin with, and it's also a state that can go away.
That's
this
right.
condition.
That's
so
right.
it's and so that's a pretty tough one. But we have had pretty amazing results in actually identifying mild cognitive impairment with multimodal technologies, and it was just being published, right? Can you tell us a little bit about this?
Yeah, absolutely. I'd love to. So we've recently published, and in fact, this is coming out this will hopefully come out soon in a journal soon as well. This very nice study of mild cognitive impairment and testing mild cognitive impairment versus healthy controls in a study with the Department of Veterans Affairs, the VA, here in San Francisco, as well as Johnson & Johnson. So that was a very nice study because for multiple reasons, actually. Just aside from the fact that, it was looking at this idea of mild cognitive impairment using these multimodal biomarkers and the prop, we were able to demonstrate using completely digital biomarkers in a decentralized trials format that we can recruit participants, up to 200 participants, in as little as 6 weeks, which is insane if you think about it, right? In clinical trials, if you tell someone 6 weeks, I'm going to recruit people in 6 weeks, they'll be like, you're out, you're insane.
Not 6 months. You mean 6 weeks?
I mean 6 weeks. Yes, yes. Sorry, that was not a mistake on my side. It was 6 weeks, not 6 months, and not 6 years, right? So it's like, this is insane. So it's worth underlining. The other thing is, so once you finish all of that recruitment, we still see high test-retest reliability within biomarkers that we obtain. And we see that a multimodal combination of biomarkers actually does the best at distinguishing between MCI, as high as, I think, 75% accuracy, if I remember this correctly, between MCI and healthy controls, which is pretty high, especially if you account for database bias, age, all of those other factors which actually inflate your accuracies in the literature. And that's not all. The use of the prop tells us something that we would not have expected otherwise. Like, for example, what we found is that the probability of someone falling or reporting that they had problems with falls was 5 times as much in people with MCI than healthy controls, which is not something which is part of the diagnostic criteria for MCI, if you think about it, right? Why would I associate falling with my cognitive impairment? But it apparently turns out to be, important. And this is something that we are able to discover because we are asking people what bothers them the most. And we get this, like you said, this idea of the platform being suitable for both, prognostic markers, kind of saying, okay, in a phase 3 study, we prescribe these biomarkers to you, but we have a whole suite of them. So we have an exploratory side of things where we can say, okay, this is how it actually affects these various components.
Going back to how is my drug, for example,
how is my drug working?
Affecting the patient. Yeah, let's move on to the next one here. How do you monitor the severity of the condition? What approaches or metrics are used to assess the progression or severity levels over time?
Great question. Again, this depends on the disease in question, but it typically is looking at how, some machine learning models, actually, in this case, to model how these biomarker values are evolving in time. And we get a more continuous valued picture, and you can benchmark that to the discrete measurements that you make of the ALSFRS or whatever. Or if it's ALS, if it's Parkinson's or schizophrenia, it will be the UPDRS or the PANS or whatever have you. But yeah, we actually use these kinds of machine learning models, not just the biomarkers themselves. So there's several levels of machine learning that we use in our technologies, which is why AI is in the name, I guess. Modality.AI. There are several levels of AI that we use.
Yeah, and that also speaks to the next question, actually, which is about confounding factors. There's always things that may influence how you measure things, like specifically how you evaluate the impact of one disease on another, which may happen as well, or the influence of other external factors on the outcomes.
The short answer to that is stratification. So when we have this information, and this is the hardest thing, right? Getting this information wherever it is required. So as part of all our clinical trials, we request a comprehensive set of background information.
Demographics.
So almost all of our metrics account for age, account for sex, account for stage of disease progression, and in many cases, where appropriate, the confounding factors like comorbidities in terms of diseases. This is a harder one to do for like observational studies. In clinical studies, it's a little bit more controlled in clinical trials. So, but yes, this is always something that we have to look out for, factor into our models.
Yeah, I wanted to double click one thing that you mentioned. This is why there's .ai in the name, but we are being asked this all the time, right?
Yeah.
What levels or what types of AI are being used?
It's a good question. So let's think through this, right? So when you open your web browser and talk to Tina, there is a module, of course, there's a signal processing module over there to collect your speech and recognize your speech. But then you're actually talking to, and by the way, all the signal processing and machine learning speech recognition is all one form of AI, right? Facial recognition is another form of AI where it recognizes your facial expressions or essentially fits a face mesh with 478 points. All of this is AI. Talking to Tina itself is a form of AI because the entire logic, as you know very well, having worked on this for many years together, right? So this whole thing of dialogue systems is its own complete branch of AI. How do you develop intelligent virtual agents? How do I make sure that the virtual agent is responding appropriately to what I just said? All of this involves AI algorithms, reasoning, and response. Same thing for generation, whether it's text-to-speech synthesis or whether it's expression generation, smile generation, all of these are forms of AI. Not just that. So all of the models that I talked about for prediction, for prognostication, for diagnosis, for progress monitoring, all of these involve versions of machine learning, which basically gets the name because you are giving the AI a bunch of training data and you're saying, learn patterns in this training data that you can then use to predict for any unseen test data. And that's something we use day in and day out. So those are just some of the examples that come to me off the top of my head, but I'm sure there are many more.
No, absolutely. And by the way, we've run out of time. Maybe the last question for today, and thank you so much for everybody to joining us today here, which really, is a continuation of what you just talked about in terms of AI. AI is this big hype, of course. Everybody uses ChatGPT and is very much aware of the many things that are happening. But at the same time, there's these downsides, hallucinations, and there's doubts about specifically the use in these high stakes environments.
That's
like
right. That's right.
Phase 3 clinical trial, which is going to be demonstrated to the FDA. So how do you see the future unfold and the use of these technologies in these high stakes trials? Is that going to be the norm, and when?
I think so. So two things, right? Two answers to that question. One is, I think I see the proliferation of decentralized digital trials for many reasons, including but not limited to two of which I already mentioned with the MCI study. One is the rapid rate of recruitment, the rapid rate of triaging. You're able to do rapid stratification for clinical trials. We've shown that we can reduce sample sizes. We can get more robust signals, all with these kinds of multimodality. So the shift I predict is going to happen towards this kind of digital decentralized trial. But to your other point, we want to double click on privacy. We want to make sure that our assessments are interpretable, that we are not infringing on privacy of participants, that we're collecting data only that is clinically meaningful and that directly applies or tells us something which is interpretable to doctors, interpretable to patients. And that's exactly the direction I see these two going. Many people think that these two really don't mesh, but I feel that now the field is actually moving towards, this kind of merging of interpretable technologies and decentralized trials and kind of moving towards responsible biomarker development. And that's something that I would like to see more of the field doing, and looking forward to it.
Well, thanks, Vikram, for giving us this positive outlook and thank you very much for spending time with us today here. And for the audience, of course, to ask these important questions and for spending time with us. And you will hear from us in a few weeks again for the next upcoming webinar. Thank you so much. You have a good rest of the day and a good weekend.
Thank you.
Bye.
See you, everyone.